Welcome back. We finished our last video talking about substitutions or point mutations that can ultimately cause three effects in proteins. One is silent, where there's no change. Missense alters one amino acid, and nonsense is going to produce a premature stop codon. Now, if we have point mutations, what we also learned last week is that we have these things called frame shift mutations. And this is going to occur when we insert. So we actually add extra bases on accident, or we delete extra bases. We call those indels because they could be insertion or deletion mutations, where we're going to add or remove one nucleotide. So again, here's our original sequence up top. And here they all are written out. Here's going to be where our start codon in the mRNA is going to originate, and where our stop is, and then we've got our codons divided out in this DNA strand. Here's the mutated version. Remember over here, we're going to look at is the effect. So we had TAC, ATG, everything looks good up until this point. But really what we're seeing here is that I've got these two red letters where we've actually inserted two nucleotides, two bases on accident, right? So this is an example of an insertion. And you guys might be able to figure out, like, well, what's going to be the impact of this? Well, my mRNA is going to have these two extra bases, which are now going to be Na and a C. And when I go through and I'm going to set my reading frame here, you guys can see ultimately what's happened, right? Instead of having ATG, TCT, AAT, even though I haven't actually changed any bases because I've inserted some, it's going to completely throw off my reading frame. So when I go through and I actually translate all these, you can see I get methionine and tyrosine like I'm supposed to, but then I start changing that amino acid from the original, right? I start changing this amino acid from the original, and then there's a premature stop codon in there. So these are disastrous. Having insertions or deletions in our code cause some pretty big changes to occur. And these changes are called frame shift mutation because they affect a number of nucleotides, which aren't divisible by 3, right? So like they're gonna alter many bases downstream from where the original amino acid would have changed. This almost always is going to result somehow in a premature stop codon as well, these frame shift mutations. Now, do we have these other types of insertions or deletions? Yeah, there actually is. We can get this entire like 6 nucleotide deletion. So here's our original strand, right? ATG, and then it goes to CTC. ATG, and whoa, like we deleted 6 bases up here, which are just completely gone. That changes my mRNA, and that's going to lead to a change in the amino acid sequence. But really what we just did here is, you guys can see we deleted 2 amino acids, or a total of 6 letters, right? 2 codons. So in these cases, these are called in-frame. Not frame shift, but in-frame mutations, which is going to affect a number of nucleotides, which is divisible by 3. In this case, we had 6. So in these insertions and deletions, not only do we have frame shift mutations, we also have in-frame mutations as well. Okay, so here's a review of how mutations can impact regions just in the coding part of the gene. Remember, we'll talk about enhancers and promoters later on. This table is a nice review for you guys, right? So we've got these substitutions or point mutations. The mutation effect could be silent, missense, or nonsense. And then the amino acid consequence is going to be the same amino acid, a different, or a premature stop codon. Okay, the next ones are what we call indels. And those can lead to frame shift mutations, which are some combination of all or some of the above, right? They're crazy. They wreak havoc. The other type of indel is an in-frame mutation, where we have addition or subtractions of amino acids. Okay, so there's a review of everything that we just discussed. Now, I want to put, there's one extra note that I want to make here. Frame shift mutations, we're not going to categorize those as causing silent, missense, or nonsense. Those we reserve for the substitutions or the point mutations. And that's because they frequently cause all of these to happen downstream. Okay, so that's an important note to make. Now let's look at this as in terms of like real impact in a disease or a disease of interest. This disease is called sickle cell disease, which many of us have probably heard about. This is caused by a change in the beta hemoglobin mutation, and that's going to cause a single nucleotide change resulting in a single amino acid change for that protein. So this is an example of what type of mutation? This is a missense mutation. It's a substitution, right? So you guys can see here, this is the sequence of the amino acids. And if we look down at position 6, there's a glutamine that's made in normal hemoglobin. Well, with one point mutation, what actually happens is instead of a glutamine being made, a valine is made. And when that valine is made, you can see this is the normal structure of hemoglobin. the protein that carries oxygen around in our red blood cells. In sickle cell hemoglobin, the shape of this beta unit, see how it's supposed to look up here? This shape is going to change, and that's due to a little bit of biochemistry of exactly why that occurs. For us, what we really need to know is that the shape changes depending on the change of the amino acid sequence. So you guys can see that shape change. Now, the function of that is that molecules under normal circumstances do not associate with one another. Each carries oxygen and everything works fine. And then you get this shape in your red blood cells. In this case, the molecules are actually going to crystallize and form into a fiber because of this shape. So the capacity to carry oxygen is ultimately reduced. So instead of having these nice separate domains, these are going to stick together and crystallize. And when that happens, we get these weird shaped or sickle shaped red blood cells. It's ultimately caused by one single base change. So what you guys can see here is this is the code that codes for normal beta hemoglobin up here on top, written 5' to 3'. And then the sequence below it is not the complementary sequence, it's the mutant or the sickle cell hemoglobin. And you can see all of the letters remain the exact same except for this one that's highlighted here in green. There's a single change from an A to a T. So instead of making a glutamine, it's going to make a valine. And it has this big of an impact on the structure of the protein, which can lead to a disease, right? And that's what we want to look at. In terms of comparison of the amino acid sequence, well, here it is, right? These are just the abbreviations of the amino acids, the one-letter abbreviation of the amino acid, and there's our one amino acid change, right? So this one substitution, this one point mutation can have a big impact on whether or not somebody actually has a disease. In the last part of today's assignment, what I'm going to have you guys go ahead and do is you're going to learn how to look this gene up and how we could actually use it in order to study, right? So what we need to do is we need to figure out how can we gain access to all these sequences if we want to compare our sequence, for example, to what the normal sequence is. So in this next video, I'm going to show you guys how do we actually look for a normal sequence. And this is all hosted in the same spot, and it's all public access. So we can look up these gene sequences, and that's what the next video is. The next thing that we would need to be able to do to figure out whether or not we actually had a disease is we'd have to be able to sequence our own DNA, right? And that's what we're going to be doing in lab. So I'm going to show you guys two key things here. One, today, how do we go find what this sequence is? And then two, in lab, what we're going to be doing is how can we actually make our own sequence? How can we pull our DNA out of our cells and make a copy of one gene and then sequence it so we can compare it to what it normally would be? And then can we use that to diagnose ourselves with certain conditions? We can. So that's what we're going to be doing in the lab. We're not going to use the sickle cell allele because that's a little gnarly, and I don't think I really want to look at that in class and have any crises. So instead, we're going to look at something a little bit more benign. But I'll teach you guys in this next video how do we look up the beta hemoglobin gene, and then we can do that same process to look up any gene that we're actually interested in.